Among the strangest discoveries in modern biology is that certain animals can persist indefinitely in the complete absence of oxygen. In 2010, the Italian zoologist Roberto Danovaro and his colleagues reported the existence of Loriciferans, minute marine invertebrates recovered from the hypersaline brine pools of the Mediterranean's Lottolonte Basin, whose entire life cycle unfolded without molecular oxygen. Years later, in 2020, Stephen Atkinson and Dorothee Huchon identified the salmon parasite Henneguya salminicola, a cnidarian relative of the jellyfish, whose genome revealed no trace of the mitochondrial machinery that animals conventionally use for aerobic respiration. These organisms were not merely tolerating short episodes of deprivation, as some vertebrates can during hibernation, but were existing permanently in oxygen-free conditions. Their cells had abandoned aerobic metabolism altogether, relying instead on alternative biochemical pathways. Placed against the human condition, the contrast is striking. A healthy adult deprived of oxygen loses consciousness in less than 2 minutes. Permanent brain injury is likely within 4, and death typically follows soon thereafter. Despite our evolutionary sophistication, our tissues remain utterly dependent on a continuous oxygen supply. Oxygen fuels every aspect of our biology: consciousness, muscular work, even the maintenance of temperature. Remove it briefly, and the integrated architecture of our physiology unravels with alarming speed. Yet oxygen is not the benign life-giving substance it is often portrayed to be. At a chemical level, it is a highly reactive molecule capable of damaging DNA, proteins, and lipids through uncontrolled oxidation. The oxygen that enables efficient energy production in mitochondria also produces reactive oxygen species, ROS, molecular fragments that corrode biological structures if not tightly regulated. This dual nature, essential to metabolism, yet inherently toxic, has made oxygen a paradoxical cornerstone of animal life. This paradox raises a fundamental scientific question. Why do some animals dispense with oxygen altogether, whereas others, including humans, are entirely dependent on it? To address it requires examining not only how oxygen sustains complex life, but also how life evolved before oxygen was present in Earth's atmosphere. The answer lies in the long co-history of biology and the planet's changing environment, a narrative that stretches back billions of years. The reliance of humans on oxygen cannot be separated from the architecture of our cells. Each mitochondrion acts as a microscopic combustion engine, taking in oxygen molecules that serve as the terminal electron acceptors in the respiratory chain. By accepting these electrons, oxygen allows the stepwise release of energy from glucose and fatty acids, channeling it into adenosine triphosphate, ATP, the universal currency of cellular energy. Without oxygen, the electron transport chain grinds to a halt, ATP production collapses, and the tissues with the highest energy requirements, particularly neurons in the cerebral cortex, cease functioning almost immediately. By contrast, organisms like Loriciferans or H. salminicola have restructured their biochemistry away from this oxygen-centered architecture. Instead of mitochondria, they rely on organelles such as hydrogenosomes or mitosomes, which trace their evolutionary origins to mitochondria but operate without oxygen. These modified structures enable forms of anaerobic respiration or fermentation, allowing cells to generate limited ATP through pathways that do not require molecular oxygen at all. The efficiency is lower, but the stability in an anoxic environment outweighs the disadvantages. For humans, the paradox deepens further because oxygen has not always been part of the biosphere. For more than 1 billion years after life emerged, Earth's oceans and atmosphere were devoid of oxygen. Early organisms survived through chemotrophic metabolisms, exploiting hydrogen, sulfur, or iron as sources of energy. When cyanobacteria began releasing oxygen as a byproduct of photosynthesis some 2.4 billion years ago, they triggered what geologists call the Great Oxidation Event. For anaerobic organisms, oxygen was initially a deadly poison. Massive die-offs reshaped microbial ecosystems, while others adapted by incorporating oxygen into their metabolic systems. This evolutionary catastrophe and opportunity set the stage for complex multicellular organisms, including ourselves, that now require oxygen absolutely. Understanding this trajectory explains why the absence of oxygen spells immediate catastrophe for humans, but represents normalcy for some microscopic animals. It is not that oxygen is universally required for life, but rather that certain evolutionary paths, including the one that led to mammals, became irreversibly tied to it. The efficiency of oxygen-based metabolism enabled higher energy outputs, larger bodies, and complex nervous systems. But this efficiency came at the cost of dependence. Where loriciferans or parasitic cnidarians can live stably in oxygen-free sediments or within their hosts, humans are locked into a system where the absence of oxygen destabilizes every vital process within minutes. The paradox, therefore, is not only chemical but evolutionary. Oxygen gives us the capacity for thought, movement, and biological complexity, but it also imprisons us in a fragile state where interruption of supply is catastrophic. The comparison with oxygen-free animals forces us to consider not merely how oxygen sustains us, but also why we cannot imagine life without it when, in fact, life on Earth long preceded its presence. It is within this frame, oxygen as both indispensable and hazardous, that we can begin to examine why humans have no capacity to dispense with it, while other animals not only endure without oxygen but thrive in its absence. The exploration must now turn to the biological mechanics that make oxygen indispensable for our own species. The human organism is constructed around a relentless demand for energy, and oxygen lies at the core of how that energy is extracted. Every cell maintains a complex interplay between macromolecules, ions, and membranes, yet the common denominator of their functioning is a need for adenosine triphosphate, or ATP. This molecule, discovered by Carl Lohmann in 1929 and later characterized in detail by Fritz Lipmann and colleagues, acts as the immediate energy reservoir for processes as varied as muscle contraction, DNA replication, and synaptic transmission. The sheer quantity of ATP required is extraordinary. Estimates suggest that a resting adult human hydrolyzes an amount of ATP each day roughly equivalent to their own body weight. To sustain such turnover, cells must maintain a highly efficient system of energy generation, and aerobic respiration provides precisely that efficiency. The mitochondrion, the organelle responsible for aerobic respiration, operates by channeling electrons derived from nutrients into an electron transport chain embedded within the inner mitochondrial membrane. The Nobel Prize-winning work of Peter Mitchell in the 1960s demonstrated how this chain couples electron transfer to the pumping of protons across the membrane, generating an electrochemical gradient. This gradient, in turn, powers ATP synthase, a rotary enzyme that produces ATP in vast quantities with remarkable efficiency. The indispensable role of oxygen arises at the terminal point of this chain. Oxygen accepts the electrons after they have passed through complexes I to IV, combining with protons to form water. Without this final electron acceptor, the chain stalls, proton pumping ceases, and ATP synthesis collapses. Nowhere is this dependence more apparent than in the brain. Although constituting only about 2% of body mass, the human brain consumes nearly 20% of the oxygen taken in at rest. This disproportionate demand is tied to the maintenance of neuronal signaling. Neurons sustain steep ion gradients across their membranes, expelling sodium and importing potassium through the ATP-powered sodium-potassium pump. Each action potential, the fundamental signal of the nervous system, depends on these gradients. When oxygen supply falters, ATP levels decline, pumps fail, ion homeostasis collapses, and neurons depolarize uncontrollably, leading to excitotoxic damage mediated by uncontrolled release of glutamate. Histological studies of hypoxic brains demonstrate how rapidly such damage manifests. Irreversible injury to hippocampal neurons can occur after as little as 5 minutes without oxygen. The heart displays a parallel vulnerability. Cardiomyocytes rely heavily on oxidative phosphorylation, deriving more than 90% of their ATP from oxygen-dependent pathways. During cardiac arrest, when circulation halts, oxygen delivery ceases, and myocardial cells switch briefly to anaerobic glycolysis. Yet this produces only a fraction of the required ATP, and lactic acid accumulation leads to acidosis, impairing contractile proteins and ion channels. If circulation is not restored quickly, the structural integrity of cardiac tissue deteriorates. Experimental models in dogs and primates during the mid-20th century established that functional recovery of the heart declines steeply with every minute of ischemia, underscoring the extreme oxygen dependence of this organ. The lungs and circulatory system have evolved elaborate mechanisms to safeguard delivery. Hemoglobin, identified in red blood cells by Felix Hoppe in the 19th century, binds oxygen with high affinity in the lungs and releases it in peripheral tissues according to partial pressure gradients. Its sigmoidal dissociation curve, first described by Christian Bohr in 1904, ensures both loading in oxygen-rich environments and unloading in oxygen-poor tissues. Yet even with such efficiency, oxygen availability is finely balanced. The human body stores remarkably little free oxygen, relying instead on continuous intake. Pulmonary reserve is measured in minutes, which is why airway obstruction or cardiac arrest results in such swift collapse of function. In contrast to species capable of withstanding prolonged hypoxia, humans possess no biochemical bypass to substitute for oxygen in energy metabolism. The glycolytic pathway can produce ATP anaerobically, but its yield is minimal. 2 molecules of ATP per glucose, compared with approximately 32 produced in the presence of oxygen. Moreover, glycolysis generates pyruvate and subsequently lactate, leading to rapid acidification of the cytoplasm and extracellular space. Accumulating hydrogen ions disrupt enzyme function and impair cellular homeostasis. Human cells, especially those of the central nervous system, are not equipped to buffer such acidosis for long durations. This dependency reflects an evolutionary trajectory in which efficiency was favored over flexibility. As multicellular organisms grew larger and developed specialized tissues, high-yield energy production became indispensable. Complex behaviors, rapid muscular responses, and large nervous systems demanded an energy throughput that only aerobic metabolism could provide. Once integrated into such a system, the capacity to revert to alternative pathways was largely lost. The enzymes and organelles required for sustained anaerobic metabolism were gradually abandoned, leaving only the vestigial glycolytic route as an emergency measure. The fragility of this arrangement is evident in medical contexts. Hypoxic, ischemic encephalopathy in newborns, high-altitude sickness in mountaineers, and myocardial infarction in adults all stem from interruptions in oxygen delivery. Each case illustrates the narrow margins within which oxygen supply must be maintained. Clinicians employ hyperbaric chambers, ventilatory support, and extracorporeal membrane oxygenation precisely because no intrinsic mechanism allows humans to dispense with oxygen, even temporarily, without severe consequences. The biochemical necessity of oxygen, then, is not merely a feature of respiration but a structural principle of human life. Every heartbeat, every synaptic impulse, every muscle contraction depends on a continuous stream of ATP derived from oxidative phosphorylation. The human body is not designed for flexibility in this regard, but for throughput and precision. Oxygen sits at the fulcrum of this system, simultaneously enabling its function and defining its vulnerability. What becomes striking when viewed against this backdrop is that Earth did not always contain oxygen in any appreciable concentration. The dominance of aerobic metabolism, which today feels indispensable, is in geological terms a relatively recent development. Understanding why oxygen became embedded so deeply in human physiology requires examining the vast shifts in planetary chemistry that altered the course of life. The reliance of complex organisms on oxygen is inseparable from Earth's transformation into an oxidizing planet. For the first 2 billion years of geological history, oxygen was absent from the atmosphere and oceans, and life developed under strictly anoxic conditions. The appearance of oxygen was not gradual background noise but a cataclysmic event, the Great Oxidation Event, beginning roughly 2.4 billion years ago. Evidence for this transition is recorded in ancient rocks, where banded iron formations, alternating layers of iron-rich minerals and silica, testify to the sudden availability of oxygen that combined with dissolved iron in the oceans, precipitating massive deposits. These structures, widespread in Precambrian strata, were first systematically analyzed in the 20th century by geologists such as Preston Cloud, who linked their formation to the earliest large-scale oxygenation of the planet. The source of this oxygen lay in cyanobacteria, prokaryotes capable of oxygenic photosynthesis. Their metabolic machinery, using water as an electron donor and releasing oxygen as a byproduct, represented an innovation that fundamentally altered the biosphere. Fossil stromatolites, layered microbial mats still visible today in regions such as Shark Bay, Western Australia, preserve evidence of these cyanobacterial communities. Radiometric dating places some of these structures at more than 3 billion years old, implying that oxygenic photosynthesis predated the atmospheric accumulation of oxygen by several hundred million years. The delay reflected a long interval during which oxygen was consumed by reduced compounds in the oceans and crust, including iron and sulfur, before it could accumulate in the air. Once those sinks were saturated, free oxygen began to build up in the atmosphere, leading to profound ecological disruption. Anaerobic organisms, adapted to an oxygen-free environment, were suddenly confronted with a reactive molecule capable of damaging their biochemistry. The Great Oxidation Event likely triggered one of the earliest mass extinctions, wiping out vast populations of anaerobes. Molecular phylogenies suggest that lineages of archaea and bacteria that survive today in extreme anoxic environments, such as methanogens and sulfate reducers, are remnants of a once dominant biosphere pushed to the margins. Oxygen, for them, was a poison, and only through retreat into niches like hydrothermal vents or deep sediments could they persist. Geochemical signatures support the magnitude of this transformation. Sulfur isotope anomalies, detectable in Archean rocks older than 2.4 billion years, disappear afterward, reflecting the rise of atmospheric oxygen to levels sufficient to alter photochemistry. Paleosols, or ancient soils preserved in rock, also indicate a shift. Reduced iron minerals disappear, replaced by oxidized forms, marking the change from an anoxic to an oxic environment. These geological traces offer independent corroboration that Earth's chemistry pivoted on a planetary scale. The consequences extended beyond extinction. Oxygen enabled entirely new metabolic pathways. Aerobic respiration, with oxygen as the terminal electron acceptor, yielded far more energy than anaerobic processes such as fermentation or sulfate reduction. This energy surplus allowed cells to expand in size, diversify their structures, and support more demanding processes. Endosymbiotic theory, articulated in modern form by Lynn Margulis in the 1960s, proposes that the ancestors of mitochondria were once free-living aerobic bacteria engulfed by larger host cells. Their integration produced the first eukaryotic cells, capable of exploiting oxygen to sustain higher energy budgets. The timing coincides with the oxygenation of the planet, suggesting a direct causal link between environmental change and cellular innovation. The rise of oxygen also altered Earth's climate. Methane, a potent greenhouse gas produced abundantly by early methanogens, reacted with the accumulating oxygen, reducing its atmospheric concentration. This chemical shift has been linked to the Paleoproterozoic glaciations, sometimes referred to as snowball Earth episodes, during which ice sheets may have extended to near-equatorial latitudes. These glaciations represent the first known instances of oxygen reshaping not only biology but planetary climate stability itself. Once oxygen had established a foothold in the atmosphere, it continued to fluctuate for nearly 2 billion years. Levels likely remained low, perhaps 1 to 10% of present atmospheric concentration, until the Neoproterozoic era. Geological proxies, including chromium isotope ratios and organic biomarkers, suggest that only around 800 million years ago did oxygen reach levels comparable to those that could sustain large multicellular organisms. Shortly afterward, the fossil record shows the emergence of the earliest animals, preserved in the Ediacaran biota. The coincidence between oxygen rise and animal diversification has long been debated, with some researchers arguing that oxygen availability provided the ecological room for complexity, while others point to developmental innovations independent of oxygen concentration. Yet the timing is striking. Complex tissues, nervous systems, and active predation require sustained energy throughput that anaerobic metabolism cannot deliver. The spread of oxygen-rich oceans in the late Precambrian provided the environmental canvas upon which these biological innovations could unfold. The Cambrian explosion, beginning around 541 million years ago, further illustrates the link. Within tens of millions of years, virtually every major animal phylum appeared, a diversification often associated with oxygen levels crossing critical thresholds. The Great Oxidation Event and its aftermath, then, did more than introduce a new gas into the air. It reordered the possibilities of biology, eliminating much of the anaerobic biosphere while opening opportunities for energy-intensive life. The trajectory that would eventually produce human dependence on oxygen originated in this planetary shift, binding metabolism to atmospheric chemistry in a way that left no path of return. The geological record, however, also reminds us that oxygen was never universally accepted by life. Even as animals emerged, large portions of the oceans remained anoxic, and microbial communities thrived in these refuges, maintaining metabolisms based on sulfur or methane. This duality, oxygen-rich habitats alongside oxygen-free zones, continued for hundreds of millions of years, shaping evolutionary strategies on both sides. The persistence of these anaerobic environments raises questions about how life maintained its foothold before oxygen became widespread, and what forms of metabolism sustained those ancient worlds. The world that nurtured the earliest organisms bore little resemblance to the modern Earth. Its oceans were chemically reducing, filled with dissolved iron, sulfides, and methane, and its atmosphere contained virtually no molecular oxygen. Under such conditions, the first metabolic strategies that evolved were tailored to exploit the available chemical gradients. Instead of relying on oxygen as a terminal electron acceptor, early cells turned to alternatives such as sulfate, nitrate, carbon dioxide, and even elemental sulfur or iron. These pathways, collectively referred to as anaerobic metabolisms, shaped the foundation of life for more than 1 billion years. Archaeological traces of this ancient chemistry are found in isotopic signatures preserved in rocks older than 3 billion years. Sulfur isotope ratios, for example, reveal microbial activity consistent with sulfate reduction, indicating that microorganisms were already capable of coupling the oxidation of organic matter or hydrogen to the reduction of sulfate long before oxygen entered the scene. The study of ancient banded iron formations points to microbial iron reduction and oxidation processes, with ferric and ferrous ion cycling driven entirely without oxygen. Such evidence shows that biology exploited Earth's mineral and chemical wealth with remarkable ingenuity. Modern representatives of these early strategies can still be found in environments that mimic primordial conditions. Hydrothermal vents along mid-ocean ridges, first directly observed in 1977 near the Galapagos Rift, host dense microbial communities thriving on hydrogen sulfide and methane emissions. These archaea and bacteria oxidize reduced sulfur or hydrogen while reducing sulfate or nitrate, generating energy without oxygen. Some of these organisms inhabit temperatures exceeding 100 degrees Celsius, challenging traditional definitions of habitability. Their existence underscores that life before oxygen was not primitive in a simplistic sense, but already diverse, resilient, and capable of biochemical sophistication. Methanogenesis represents another cornerstone of pre-oxygen biology. Certain archaea, such as Methanobrevibacter smithii in the human gut or Methanococcus jenascii in hydrothermal vents, generate methane by reducing carbon dioxide with hydrogen. This pathway not only provides energy but also releases methane into the environment, a gas that may have helped maintain Earth's surface temperatures under the faint young sun by strengthening the greenhouse effect. The presence of widespread methanogenic activity in Earth's early biosphere links microbial metabolism directly to planetary climate regulation, long before oxygen shaped the atmosphere. Nitrogen cycling also emerged under anoxic conditions. Denitrification, the reduction of nitrate to nitrogen gas, supplied energy to early microbes and influenced the composition of the oceans and atmosphere. Some evidence suggests that biological nitrogen fixation, converting atmospheric nitrogen into bioavailable ammonium, was already present by 3.2 billion years ago. These cycles formed the ecological scaffolding on which later oxygen-dependent processes would build, ensuring that carbon, nitrogen, and sulfur were already circulating through interconnected microbial networks. The architecture of cells reflected these conditions. Without oxygen, mitochondria could not yet exist. Instead, energy-producing machinery resided in the cytoplasmic membrane, where enzymes drove electron transfers across lipid bilayers to create proton gradients. ATP synthase, the rotary enzyme later co-opted by mitochondria and chloroplasts, is thought to have evolved during this time, coupling proton flow to ATP production in strictly anaerobic contexts. The continuity of this enzyme from the earliest cells to present-day organisms highlights how fundamental bioenergetic principles were established in the absence of oxygen. The endurance of anaerobic ecosystems into the present illustrates that the pre-oxygen world has never truly vanished. In wetlands, swamps, and waterlogged soils, where oxygen diffusion is limited, communities of sulfate-reducing bacteria and methanogenic archaea dominate. In deep subsurface sediments, sealed off from surface oxygen for millions of years, microbial life persists at extremely slow metabolic rates, sometimes dividing only once per century. Isotopic and genomic studies of these communities reveal adaptations for life under chronic energy limitation, reminding us that oxygen-intensive metabolism is not the universal standard but one branch of a much broader evolutionary spectrum. The survival of these lineages also challenges assumptions about complexity. While oxygen allowed for energy-intensive multicellular life, many anaerobes display elaborate cellular structures and regulatory systems. Some archaea, such as members of the Asgard group discovered in 2015 in marine sediments off the coast of Norway, possess cytoskeletal proteins and intracellular compartments once thought to be restricted to eukaryotes. These discoveries suggest that the building blocks of complexity were already present in anaerobic organisms, and may have played a role in the eventual emergence of oxygen-using cells. From a biochemical perspective, anaerobic life avoids one of the key hazards of oxygen metabolism: oxidative stress. By not using oxygen, these organisms sidestep the formation of reactive oxygen species that can damage cellular components. Their vulnerability lies instead in competition for limited resources and the low efficiency of their energy pathways. Yet in environments where oxygen never penetrates, efficiency is less important than stability. Anaerobic metabolisms endure precisely because they are matched to the chemical landscapes that still exist on Earth. The persistence of oxygen-free life illuminates a critical point about biological evolution. Life did not progress in a linear trajectory from anaerobic simplicity to aerobic complexity. Instead, multiple strategies have coexisted for billions of years, each adapted to particular geochemical settings. Oxygen created new possibilities, but did not erase the old ones. The biosphere today remains a mosaic of these metabolic traditions, from deep-sea methanogens to surface-dwelling mammals, each lineage bearing witness to a different stage of Earth's chemical evolution. Yet the question that presses forward is how multicellular organisms, creatures far larger than bacteria or archaea, could establish themselves in oxygen-free environments when oxygen appeared to become the dominant driver of biological expansion. To uncover that development requires examining the rare but extraordinary cases in which animals themselves abandoned the oxygen pathway and charted a different evolutionary course. For much of the 20th century, biologists regarded oxygen as an absolute requirement for animals. While certain vertebrates could survive temporary oxygen shortages by suppressing metabolism, the prevailing assumption was that no animal could complete its life cycle entirely without oxygen. That assumption was overturned in the early 21st century with the discovery of multicellular organisms thriving in permanently anoxic environments. Their existence not only challenged entrenched views of animal physiology, but also forced a reconsideration of what it means to be an animal at all. The most striking examples came from the hypersaline basins of the deep Mediterranean Sea. These basins, located several kilometers below the surface, are filled with dense brines formed by the dissolution of ancient evaporite deposits. The brines are so saline that they create a physical barrier, preventing mixing with overlying oxygenated seawater. Conditions inside these basins are extreme. No oxygen, high pressure, salinity several times greater than seawater, and high concentrations of hydrogen sulfide. For decades, such habitats were thought to be the preserve of microbes alone. In 2010, a team led by Roberto Danovaro of the Polytechnic University of Marseille sampled sediments from the Lottolonte, Urania, and Discovery basins. Using remotely operated vehicles, they retrieved cores of anoxic mud sealed from oxygen contamination. Microscopic examination revealed the presence of Loriciferans, tiny marine invertebrates belonging to a phylum first described only in 1983. Under electron microscopy, these animals displayed intact tissues, eggs, and organelles, suggesting not accidental burial but active, ongoing life. Crucially, their cells contained unusual structures resembling hydrogenosomes, organelles known from anaerobic protists that generate ATP without oxygen. No mitochondria were visible, and genetic studies indicated that the classical aerobic machinery had been abandoned. These Loriciferans represented the first confirmed animals with a metabolism adapted to permanent anoxia. Equally unexpected was the discovery in 2020 of Henneguya Salminicola, a cnidarian parasite of salmon. This organism, related to jellyfish and corals, has a radically simplified body plan consisting of just a few cell types. Genome sequencing by Dorothee, Huchon and colleagues at Tel Aviv University revealed the absence of mitochondrial genes required for aerobic respiration. Instead, the parasite retained mitosomes, organelles derived from mitochondria but incapable of generating ATP through oxygen use. How H. salminicola meets its energy demands remains a subject of investigation, with hypotheses pointing to direct absorption of ATP from host tissues or reliance on anaerobic biochemical pathways. The striking feature is that an animal lineage, albeit parasitic and reduced, has entirely discarded oxygen metabolism. These findings shocked the biological community because they contradicted the assumption that mitochondria, and with them oxygen-based respiration, were indispensable hallmarks of animal life. The prevailing textbook view, grounded in decades of comparative physiology, was that animals owed their complexity to the energy yield of aerobic metabolism. The Loriciferans and parasitic cnidarians demonstrated that exceptions exist, and that under certain ecological pressures, even animals can abandon oxygen altogether. The mechanisms underlying this independence vary. Hydrogenosomes, found in Loriciferans and numerous anaerobic protists, function by coupling substrate-level phosphorylation to the production of hydrogen gas, often in symbiosis with hydrogen-consuming microbes. Mitosomes, as in H. salminicola, have even fewer functions, retaining roles in iron, sulfur cluster assembly but not in energy generation. These modified organelles are evolutionary relics of mitochondria, reshaped to fit environments where oxygen is absent. Their existence underscores the plasticity of eukaryotic cell biology. Structures once dedicated to oxygen-dependent processes can be retooled for survival under entirely different conditions. The ecological niches these animals occupy are instructive. Hypersaline basins, parasitic habitats within host tissues, and other isolated anoxic environments represent refuges where oxygen cannot penetrate. In such settings, the selective advantage of aerobic metabolism vanishes, while the liabilities of oxygen toxicity become irrelevant. Animals that survive here are not less adapted but differently adapted, having exchanged efficiency for compatibility with their surroundings. The presence of oxygen-free animals also reframes questions about the limits of metazoan evolution. If even a small number of lineages have dispensed with oxygen, it implies that the evolutionary constraints on animal metabolism are broader than once believed. The number of such lineages may be underestimated, since exploration of anoxic environments remains limited by technical challenges. Deep-sea brine pools, subterranean aquifers, and oxygen-depleted sediments may harbor additional examples, awaiting discovery by molecular and microscopic analysis. The shock within the scientific community was not only conceptual but methodological. Detecting active life in permanently anoxic sediments required extraordinary precautions against contamination. Critics initially questioned whether Loriciferans were alive when recovered, or whether they had simply been transported there post-mortem. Subsequent evidence of reproduction and metabolically active organelles silenced most doubts, but the debate highlighted how radical the idea seemed within established frameworks of zoology. The case of H. Salminicola was strengthened by genomic data, removing ambiguity and demonstrating unequivocally that an animal can survive without mitochondrial respiration. What remains unresolved is the full biochemical picture of how such animals manage energy homeostasis in the absence of oxygen. Detailed proteomic and metabolomic studies are still ongoing, aiming to identify the precise pathways involved. Whether these animals rely entirely on their own anaerobic enzymes or draw heavily on symbiotic microbes is an open question. The blurred line between self-sufficiency and dependence on microbial partners complicates the definition of what it means for an animal to live without oxygen. Yet the fact that they do persist, reproduce, and evolve under these constraints stands beyond doubt. If oxygen-free animals exist, even in limited niches, the definition of what is biologically possible expands. It compels a reconsideration of the dogma that complexity requires oxygen, and it draws attention to the physiological limits of other organisms that cannot dispense with it. For humans, whose nervous and muscular systems collapse within minutes of deprivation, the comparison is stark. Understanding why some animals can navigate life without oxygen, while we cannot, requires turning toward the fragility of mammalian physiology, where oxygen dependency reaches its most unforgiving form. Among all organs in the human body, the brain stands as the most uncompromising in its reliance on oxygen. Its dependence is not incidental, but built into the very architecture of neural signaling. Each neuron, elongated and branched into complex networks, requires a steep and continuous flow of energy to maintain electrical excitability. The sodium, potassium ATPase, an enzyme embedded in the neuronal membrane, consumes an estimated 50 to 60% of the brain's entire energy budget, restoring ion gradients after every impulse. This relentless demand means that the brain's functional stability is inseparable from a steady supply of oxygen. The vulnerability of neurons to oxygen deprivation was first characterized in experimental detail in the early 20th century. In 1911, Leonard Hill demonstrated that dogs exposed to low oxygen conditions lost consciousness rapidly, and his observations helped establish the principle that neuronal failure precedes failure of other organs. Later studies, using electroencephalography in humans during controlled hypoxia, confirmed that brain activity declines within seconds when oxygen is withdrawn. The loss of consciousness experienced by pilots at high altitude without supplemental oxygen is a manifestation of this sensitivity. Unlike muscle cells, which can endure anaerobic glycolysis for short periods, neurons have no capacity for sustained anaerobic energy production. The biochemical reason lies in the mismatch between demand and yield. Glycolysis alone generates only 2 ATP molecules per glucose, insufficient to sustain the high-frequency firing of cortical neurons. Furthermore, the accumulation of lactate during anaerobic metabolism disrupts pH balance in neural tissue. Unlike skeletal muscle, which tolerates short-term acidosis during intense exercise, the brain lacks buffering capacity. Even small deviations in pH interfere with neurotransmitter release, receptor sensitivity, and ion channel function. Experimental perfusions of isolated brain slices with lactate-rich solutions show rapid deterioration of synaptic transmission, underscoring how intolerant the brain is to anaerobic conditions. Ischemic injury illustrates the cascade of damage triggered by oxygen loss. Within 1 minute of severe hypoxia, ATP depletion leads to failure of ion pumps, depolarization of neuronal membranes, and uncontrolled release of glutamate into synapses. Excess glutamate overactivates NMDA receptors, allowing toxic influxes of calcium ions. This excitotoxicity activates proteases, lipases, and endonucleases, degrading essential cellular components. Mitochondria, overwhelmed by calcium, generate reactive oxygen species when oxygen is reintroduced, exacerbating injury. The process, documented extensively in stroke pathology, demonstrates that neurons are not merely passive victims of oxygen deprivation but active participants in their own destruction once energy balance collapses. Other mammals display similar constraints, though with varying degrees of tolerance. Laboratory studies in rodents show irreversible neuronal injury after 5 to 10 minutes of complete ischemia, with hippocampal and cortical neurons being the most vulnerable. Larger mammals, including primates, show comparable thresholds. Hypoxia-inducible factors, HIFs, transcription factors that upregulate survival pathways under low oxygen, provide some protection, but their effects are limited to gradual decreases in oxygen availability. Acute anoxia overwhelms these adaptive mechanisms. Humans at high altitude exhibit increased expression of HIF-regulated genes, stimulating angiogenesis and red blood cell production, yet these responses cannot sustain brain function without oxygen altogether. The evolutionary explanation for this fragility lies in the trade-off between energy efficiency and flexibility. The expansion of the neocortex in hominins required enormous energy throughput. Comparative studies estimate that the human brain consumes about 20 watts at rest, accounting for nearly 1/5 of basal metabolic rate. Such a system could only be sustained by oxidative phosphorylation, the most energy-dense pathway available. In exchange for high capacity, evolutionary selection abandoned the ability to function under anaerobic conditions. The result was a nervous system capable of complex cognition, language, and abstract thought, but incapable of tolerating interruption in oxygen supply. Experiments attempting to induce tolerance have revealed the rigid limits of this dependence. Hypothermia, used clinically in cardiac surgery and neonatal encephalopathy, slows metabolic rate and reduces oxygen consumption, thereby prolonging neuronal survival under ischemia. Yet even under cooled conditions, neurons cannot dispense with oxygen indefinitely. They merely delay the onset of irreversible injury. Similarly, pharmacological suppression of neuronal activity with barbiturates lowers demand but does not confer independence. These interventions highlight that oxygen is not simply a fuel, but an integral part of the brain's metabolic machinery, woven into every aspect of its design. The heart and skeletal muscles, though also reliant on oxygen, possess fallback strategies that neurons lack. Cardiomyocytes can shift briefly to anaerobic glycolysis, and skeletal muscle fibers are accustomed to fluctuating oxygen availability during exertion. By contrast, The brain evolved under selection pressures that rewarded continuous high-frequency signaling rather than intermittent tolerance. Any lapse in neuronal activity would have compromised the complex behaviors and social interactions that define survival. Over evolutionary timescales, this locked the brain into its present vulnerability. Attempts to identify animals with oxygen-tolerant nervous systems reveal instructive contrasts. Certain freshwater turtles, such as Trachemys scripta elegans, can survive months of anoxia under ice by depressing neuronal activity and releasing inhibitory neurotransmitters that silence much of the brain. Carp employ similar strategies, producing ethanol as an end product of anaerobic glycolysis to avoid lactate buildup. These adaptations, however, come at the cost of reduced responsiveness. The human brain, optimized for constant processing and rapid reaction, lacks such flexibility. Its specialization for speed and complexity precluded the adoption of energy-saving modes that could allow anoxia tolerance. The paradox is that the very organ that defines human uniqueness is also the least capable of coping with oxygen loss. Medical science can delay damage with cooling, drugs, or mechanical intervention, but cannot rewrite the fundamental relationship between neurons and oxygen. This raises questions about whether the human body could ever evolve or be engineered to acquire tolerance, or whether our dependency represents an evolutionary dead end from which no adaptation is possible. The answer requires examining species that have managed to survive low oxygen conditions, not by abandoning oxygen altogether, but by stretching its limits in remarkable ways. While the human nervous system disintegrates quickly when deprived of oxygen, some vertebrates have evolved extraordinary strategies to tolerate prolonged hypoxia or even complete anoxia. These animals do not dispense with oxygen as a metabolic principle, but they can suspend their dependence for remarkable lengths of time. The distinction between tolerance and independence is critical. They survive without oxygen not by abandoning it, but by entering states in which metabolic processes slow to a fraction of their normal rates. One of the most studied examples is the freshwater turtle Trachemys scripta elegans, native to North America. During winter, when lakes and ponds freeze, these turtles remain trapped beneath ice, often in water entirely cut off from atmospheric oxygen. Laboratory studies in the 1980s and 1990s, particularly those conducted by Peter Lutz and colleagues at Florida Atlantic University, demonstrated that painted turtles can survive 3 to 4 months of complete anoxia at low temperatures. Their strategy involves a profound depression of metabolic activity, reducing energy consumption by more than 90%. Neurons that under normal conditions fire continuously are silenced, and ATP turnover slows to levels that can be sustained by anaerobic glycolysis alone. This suppression of brain activity is not passive failure, but an active adaptation. Turtles release large amounts of the inhibitory neurotransmitter gamma-aminobutyric acid, GABA, which hyperpolarizes neurons and prevents uncontrolled depolarization. They also regulate potassium channel activity, stabilizing membrane potential in ways that avoid excitotoxic cascades. The result is a reversible, silent brain, in which function is preserved without the destructive overactivity typical of mammalian neurons deprived of oxygen. Another critical element of turtle survival is buffering against acidosis. Anaerobic glycolysis generates lactate, and in most animals, accumulation of this metabolite rapidly disrupts cellular function. Turtles counteract this by storing lactate in their shells, where calcium carbonate reacts with hydrogen ions to neutralize acidity. This buffering system, unique among vertebrates, allows them to tolerate lactate levels that would be lethal to mammals. Measurements of blood chemistry in overwintering turtles show lactate concentrations exceeding 200 millimoles per liter, more than 10 times those tolerated by humans. Carp and goldfish, Carassius carassius and Carassius auratus, display a different but equally remarkable adaptation. In ice-covered lakes of northern Europe and Asia, these fish endure months with little or no dissolved oxygen. Their metabolic solution is to convert pyruvate, the product of glycolysis, not into lactate but into ethanol, which diffuses harmlessly across the gills into the surrounding water. This pathway, first elucidated in the 1960s by Scandinavian researchers, prevents the buildup of lactic acid and spares the fish from acidosis. Ethanol production is energetically costly, but it represents a viable compromise that allows survival in oxygen-free conditions until spring thaw restores oxygen levels. Amphibians provide additional strategies. Frogs such as Rana sylvatica can withstand freezing solid during winter, their bodies filled with ice crystals. Although oxygen transport halts under these conditions, metabolic depression and cryoprotectants such as glucose and glycerol preserve cells until thawing occurs. Other amphibians rely on cutaneous respiration, absorbing oxygen directly through their skin, which allows survival in low-oxygen waters where pulmonary respiration is ineffective. These methods highlight the diversity of solutions available within vertebrates for coping with hypoxia. The unifying feature of these strategies is tolerance rather than independence. Turtles, carp, and amphibians do not reject oxygen as a metabolic substrate. On the contrary, their survival depends on returning to aerobic metabolism when conditions allow. Their adaptations buy time, weeks or months during which life processes continue at minimal rates, but cannot sustain indefinite existence without oxygen. This contrasts sharply with the true oxygen-free animals described earlier, whose entire biology is structured around the permanent absence of oxygen. The physiological extremes of vertebrate tolerance, nonetheless, offer insights into mechanisms that humans lack. The ability to suppress neuronal excitability, buffer metabolic acidosis, or channel glycolytic end products into less toxic compounds represents alternative pathways for maintaining viability. Experimental studies have attempted to translate these mechanisms into clinical strategies. For example, pharmacological agents that enhance GABAergic inhibition have been investigated as neuroprotective treatments for stroke, inspired by turtle anoxia resistance. Similarly, induced hypothermia, which mimics the metabolic depression of overwintering species, is now used in cardiac arrest patients to reduce brain injury. These applications illustrate how understanding natural tolerance can inform medicine, even if humans cannot replicate the full adaptations of other vertebrates. The ecological implications are equally striking. In temperate climates, winter survival under ice would be impossible without such strategies. By exploiting metabolic suppression, these animals occupy niches closed to less tolerant species, reducing competition and ensuring reproductive success when conditions improve. Evolution has not produced universal independence from oxygen in vertebrates, but it has produced a spectrum of tolerances that extend the boundaries of survival far beyond human limits. Yet these examples still leave open the question of why smaller animals, particularly invertebrates, can thrive permanently in oxygen-free habitats while larger vertebrates must rely on temporary tolerance. The answer lies partly in body size and metabolic scaling. Smaller animals with lower energy demands can persist on limited anaerobic pathways that would be insufficient for larger organisms. The diversity of such adaptations becomes visible in the oxygen-deprived sediments and soils where countless invertebrates make their living, demonstrating that tolerance, when combined with size and simplicity, can blur the line between temporary survival and permanent existence. If vertebrates display elaborate but temporary strategies to bridge periods of oxygen deprivation, many invertebrates live in habitats where oxygen is chronically scarce or entirely absent. Their adaptations are not designed for short-term survival during seasonal crises, but for daily existence in muds, swamps, and seabeds where oxygen penetrates only a few millimeters below the surface. These environments, rich in organic matter but poor in diffusion, represent some of the most widespread ecological niches on Earth. Within them, worms, mollusks, crustaceans, and insect larvae persist through combinations of anaerobic metabolism, symbiosis with microbes, and structural adaptations that minimize oxygen demand. Marine sediments are prime examples of such habitats. oxygenated water contacts the seafloor, but microbial respiration consumes oxygen so rapidly that anoxic conditions dominate below the uppermost layer. Benthic invertebrates dwelling in these sediments must either remain near the interface, where some oxygen is available, or evolve means to function without it. Polychaete worms of the genus Capitella, for instance, flourish in organically enriched, oxygen-poor muds. They rely heavily on anaerobic metabolic pathways, converting glucose into end products such as succinate and propionate. These metabolites accumulate at levels that would incapacitate many animals, but polychaetes have evolved tolerance to such conditions. Certain bivalves, such as Solemia velum, employ an alternative strategy: symbiosis with sulfur-oxidizing bacteria housed within their gills. These symbionts use hydrogen sulfide present in the sediment as an energy source, producing organic compounds that nourish the host. The relationship allows the clam to thrive in habitats devoid of oxygen, effectively outsourcing its metabolism to microbial partners. Similar partnerships exist in lucinid clams and vestimentiferan tubeworms, demonstrating that invertebrates often survive in anoxia, not through their own biochemistry alone, but through intimate alliances with bacteria. In freshwater wetlands, insect larvae provide another perspective. Mosquito larvae, culex and anopheles, access surface oxygen through specialized breathing tubes, but other insects, such as certain chironomid midges, tolerate extended periods buried in oxygen-depleted mud. These larvae synthesize large quantities of hemoglobin, unusual among invertebrates, which stores and releases oxygen at extremely low concentrations. Their bright red coloration derives from this hemoglobin, earning them the common name bloodworms. In laboratory studies, chironomid larvae maintained metabolic activity at oxygen levels so low that conventional detection instruments registered near-zero concentrations, highlighting the effectiveness of their pigment adaptations. Crustaceans in anoxic environments often exploit metabolic flexibility. The brine shrimp Artemia franciscana, a model organism in developmental biology, can enter dormant cyst stages that withstand complete anoxia for years. Within these cysts, metabolic activity declines to undetectable levels, protecting the embryo until favorable conditions return. When oxygen becomes available, metabolism resumes, and development proceeds. Although dormancy differs from active life, it illustrates how invertebrates can span periods of anoxia without damage, a capacity largely absent from vertebrates. Size plays a crucial role in enabling these strategies. The smaller the organism, the lower its absolute energy demand, making survival on limited anaerobic pathways feasible. Worms, mollusks, and larvae require fewer molecules of ATP per unit time than large vertebrates, permitting them to balance energy budgets even when restricted to inefficient fermentation. This scaling principle explains why oxygen-free existence is more common among invertebrates than vertebrates. The structural simplicity of many invertebrates, with fewer energy-intensive tissues like complex nervous systems, further reduces their vulnerability. At the biochemical level, anaerobic invertebrates employ a wide array of pathways. Beyond lactic acid fermentation, they often produce volatile fatty acids such as acetate or propionate, and in some cases ethanol, to avoid excessive acidosis. The diversity of end products reflects evolutionary experimentation under oxygen-limited conditions. In the lugworm Arnicola marina, succinate is the major anaerobic end product, while in other annelids, propionate predominates. Such differences indicate that anaerobic metabolism is not a single pathway, but a collection of solutions tailored to specific environments. Research into these animals often reveals overlooked ecological consequences. By consuming organic matter under anoxic conditions, benthic invertebrates influence nutrient cycling and the chemistry of sediments. Their waste products, including hydrogen sulfide and ammonia, feed microbial processes that shape entire ecosystems. In some estuaries, Dense populations of anoxia-tolerant worms and clams drive sulfur and nitrogen cycles that determine water quality and productivity. These contributions underscore that adaptations to oxygen scarcity are not only individual survival strategies, but also integral to ecological stability. Yet the existence of oxygen-free invertebrates also raises questions about limits. How far can anaerobic metabolism sustain complexity? While worms and clams function well, they remain small and relatively simple compared to vertebrates. Their nervous systems, though functional, do not demand the high-energy throughput characteristic of mammalian brains. Their circulatory and muscular systems, though capable, operate on slower timescales. This suggests that anoxia-tolerant invertebrates thrive by embracing modest metabolic rates, rather than by developing alternatives that could support the high demands of vertebrate physiology. The persistence of such animals across diverse environments demonstrates that oxygen-free existence is not an anomaly, but a recurring theme in evolution. Their adaptations illustrate how biological systems can exploit chemical diversity to persist where others cannot. Yet their modest scale points toward a ceiling on what anoxia tolerance alone can achieve. Understanding how life at smaller scales negotiates anoxia opens the path to examining the even more radical strategies of microscopic organisms, where the rules of survival shift yet again into domains of dormancy, cryptobiosis, and extraordinary resilience. The smallest organisms often reveal the broadest repertoire of survival strategies under oxygen deprivation. Unlike vertebrates that suppress metabolism only temporarily or invertebrates that endure with modest energy demands, many microscopic life forms navigate anoxia with mechanisms so varied and resilient that they have persisted across geological epochs. Among protozoa, rotifers, and tardigrades, the boundaries of metabolic adaptation are pushed to extremes, demonstrating that the absence of oxygen can be not only tolerated but incorporated into a stable way of life. Protozoa furnish some of the most instructive examples. Parasitic lineages such as Giardia lamblia and Entamoeba histolytica inhabit the anaerobic environments of host intestines. Their cells contain mitosomes, reduced organelles derived from mitochondria, that no longer perform oxidative phosphorylation but retain functions such as iron, sulfur cluster assembly, essential for enzymatic activity. Instead of aerobic respiration, these protozoa generate ATP through substrate-level phosphorylation, often using pyruvate, ferredoxin oxidoreductase pathways that release hydrogen as a byproduct. Symbiotic associations with hydrogen-consuming methanogens remove the hydrogen, stabilizing the internal redox balance. Such metabolic networks illustrate that even single-celled eukaryotes can maintain elaborate biochemical systems entirely without oxygen. Rotifers add another dimension. Deloid rotifers, microscopic animals common in freshwater habitats, display extraordinary tolerance to desiccation and oxygen deprivation. When confronted with anoxic conditions, they enter states of dormancy in which metabolic activity declines to undetectable levels. Laboratory experiments have demonstrated that rotifers can survive years of anoxia, reviving when oxygen returns. Their genomes reveal extensive horizontal gene transfer from bacteria and fungi, suggesting that the integration of foreign genes may contribute to their resilience. The biochemical basis of their dormancy involves vitrification of cytoplasmic contents, stabilization of membranes, and the accumulation of protective sugars such as trehalose. In this suspended state, oxygen is unnecessary because all energy-consuming processes halt until reactivation. Tardigrades, often called water bears, are even more remarkable. These microanimals, discovered in the 18th century by Johann August Guz, have become icons of extremophile biology. They can withstand complete desiccation, freezing to near absolute zero, and exposure to high radiation. Their strategy under anoxia parallels their response to desiccation, entry into a cryptobiotic state known as a tun. During this process, the tardigrade contracts into a barrel-like shape, reduces water content, and shuts down metabolic activity to almost zero. Experiments by Italian zoologist Roberto Danovaro and others have shown that tardigrades in ton states can survive years without oxygen, resuming activity when rehydrated in oxygenated water. Molecular studies indicate that unique proteins, including tardigrade-specific intrinsically disordered proteins, form glass-like matrices that stabilize cellular structures, allowing life to persist in a state that is neither fully active nor dead. The ability of microorganisms to endure anoxia is not limited to short intervals. Microbial communities have been discovered in subglacial lakes sealed beneath Antarctic ice for hundreds of thousands of years, as well as in deep marine sediments more than 100 million years old. In these habitats, oxygen is absent, and metabolic rates are extraordinarily low. Cells divide only once per century, yet they maintain viability, repairing DNA damage and preserving protein function over immense timescales. Geomicrobiological investigations, such as those conducted at the Integrated Ocean Drilling Program, confirm that life can persist in conditions of extreme energy scarcity, where oxygen never plays a role. Fermentation remains a central mechanism in these settings. Organisms recycle NADH to NAD+ by transferring electrons to organic molecules, producing end products like ethanol, acetate, or hydrogen. Although the ATP yield is minimal, in energy-limited environments, efficiency is less critical than continuity. Some ciliates, such as Paramecium cordatum, harbor endosymbiotic methanogens that consume hydrogen generated during fermentation, enabling continued energy flow without oxygen. The intimacy of these associations blurs the line between host and symbiont, producing functional units that resemble miniature ecosystems within a single cell. What distinguishes microscopic organisms from larger invertebrates or vertebrates is the sheer diversity of their strategies. They can alternate between active anaerobic metabolism and cryptobiosis, partner with microbes to offload toxic byproducts, or shut down life processes entirely. This multiplicity ensures that no single environmental change can easily extinguish them. For example, when permafrost cores in Siberia were thawed, viable nematodes and protozoa revived after tens of thousands of years in suspended animation. Radiocarbon dating of the surrounding sediments confirmed their antiquity, demonstrating that anoxia combined with cryopreservation can maintain life for geological durations. The resilience of microscopic organisms under anoxia has significant implications for ecology. In soils and sediments, anaerobic protozoa regulate bacterial populations, shaping nutrient cycles. Rotifers and tardigrades play key roles in freshwater ecosystems, where their tolerance allows them to colonize ephemeral habitats that cycle between oxygen-rich and oxygen-poor states. Their survival strategies ensure continuity of ecosystems through droughts, freezes, and seasonal anoxia. On a planetary scale, they embody a principle of persistence that stands in sharp contrast to the fragility of oxygen-dependent vertebrate physiology. Yet the very versatility of these organisms also illuminates the double-edged nature of oxygen itself. While oxygen permits high energy output, it imposes vulnerability through its reactive byproducts. Microscopic organisms that avoid oxygen altogether sidestep the constant burden of oxidative stress, a cost that aerobic organisms must mitigate through complex antioxidant systems. The evolutionary trade-offs between embracing oxygen and rejecting it appear most clearly when examining how cells protect themselves from oxygen's damaging chemistry. The paradox of oxygen lies in its dual role. It is indispensable as the terminal electron acceptor in respiration, yet inherently hazardous because of its chemical reactivity. The same property that allows oxygen to capture electrons efficiently also makes it prone to forming reactive intermediates capable of attacking biological molecules. These reactive oxygen species, ROS, include superoxide anion, O2, hydrogen peroxide, H2O2, and hydroxyl radicals, AirOH. Each arises during the normal operation of the electron transport chain in mitochondria, when electrons occasionally leak from respiratory complexes I and III to molecular oxygen. Although leakage is typically only a few % of total electron flow, the cumulative effect across billions of mitochondria and trillions of cells is significant. The destructive potential of ROS stems from their tendency to oxidize macromolecules indiscriminately. DNA bases suffer oxidative lesions such as 8-oxoguanine, which can mispair during replication and introduce mutations. Proteins undergo carbonylation, altering their function and triggering degradation. Lipid peroxidation, particularly in membranes rich in polyunsaturated fatty acids, compromises structural integrity and disrupts signaling pathways. In the 1950s, Rebecca Gershman and Daniel Gilbert were among the first to propose that oxygen toxicity derived from free radical reactions, an idea later solidified by Denham Harmon's free radical theory of aging in 1956. Since then, an enormous body of experimental work has confirmed that ROS are unavoidable byproducts of aerobic metabolism. Biological systems counteract these threats with an elaborate arsenal of antioxidant defenses. Enzymes such as superoxide dismutase convert superoxide into hydrogen peroxide, which is then decomposed into water and oxygen by catalase or glutathione peroxidase. Small molecule antioxidants, including glutathione, vitamin C, and vitamin E, scavenge radicals and terminate chain reactions. Despite these safeguards, oxidative damage accumulates with age and stress, reflecting the imperfect balance between ROS generation and detoxification. Measurements of oxidative lesions in mitochondrial DNA increase steadily in older tissues, correlating with declines in respiratory efficiency. Some organisms mitigate oxygen toxicity not by defending against ROS, but by avoiding oxygen altogether. Anaerobes thrive precisely because they exclude oxygen from their metabolism, sidestepping the burden of oxidative stress. For these organisms, oxygen is not a fuel but a toxin that inactivates enzymes containing iron, sulfur clusters, or other oxygen-sensitive cofactors. Strict anaerobes, such as Clostridium species, lack the enzymatic systems to neutralize ROS and perish rapidly upon exposure. Their vulnerability underscores that aerobic life carries constant costs, and that the adoption of oxygen was never universally advantageous. The toxicity of oxygen has been most vividly demonstrated in clinical contexts. Hyperbaric oxygen therapy, while medically useful in specific cases such as decompression sickness and wound healing, can provoke seizures and pulmonary injury when administered at high pressures for extended periods. These effects arise from excessive ROS production, overwhelming antioxidant defenses. Premature infants exposed to high concentrations of oxygen in incubators sometimes develop retinopathy of prematurity, a condition where abnormal blood vessel growth damages the retina. These clinical examples reveal how narrow the margin is between beneficial oxygen levels and toxic exposure. At the cellular level, ROS also function as signaling molecules, complicating their role. Low levels of hydrogen peroxide act as second messengers in pathways regulating proliferation, immune responses, and adaptation to stress. The transcription factor NF-kappa B, for instance, is activated by ROS and orchestrates inflammatory signaling. Similarly, hypoxia-inducible factors are stabilized when ROS production shifts during low oxygen, modulating genes involved in angiogenesis and metabolism. This dual function, both damaging and regulatory, illustrates the fine line between oxygen as a poison and oxygen as a physiological signal. The evolutionary embrace of oxygen thus entailed not only exploiting its energetic yield, but also evolving mechanisms to manage its toxicity. Comparative genomics shows that superoxide dismutase enzymes are ancient, predating the full rise of oxygen in the atmosphere. Their presence in early organisms suggests that even trace oxygen levels required defensive adaptation. As oxygen concentrations increased during the Proterozoic, antioxidant systems diversified, allowing organisms to tolerate higher exposures. Yet the cost of oxidative damage was never eliminated, only managed. The consequences of this compromise extend into pathology. Neurodegenerative disorders such as Parkinson's and Alzheimer's disease feature oxidative stress as a prominent factor, with ROS contributing to protein misfolding and neuronal death. Cardiovascular diseases involve oxidative modification of low-density lipoproteins, promoting atherosclerosis. Cancer cells often exhibit elevated ROS, which both drives mutagenesis and stimulates signaling pathways that support uncontrolled growth. These examples illustrate that oxygen's toxicity is not a minor side effect, but a central force in shaping health and disease. Some animals exploit ROS intentionally as weapons. Phagocytes in the human immune system deploy an oxidative burst, releasing superoxide and hydrogen peroxide to kill engulfed bacteria. Enzymes such as myeloperoxidase amplify this effect by generating hypochlorous acid. In these contexts, oxygen toxicity becomes a tool for defense, highlighting once again its ambivalent nature: harmful when uncontrolled, useful when directed. The persistent dangers of oxygen raise an unresolved question. Why did evolution not favor more widespread alternatives that avoid oxygen altogether? The answer lies partly in the unparalleled energy yield of aerobic respiration, which supports complexity beyond the reach of anaerobic pathways. Yet this energy comes at the price of constant vigilance against oxygen's damaging chemistry. The evolutionary compromise has left organisms not only dependent on oxygen, but perpetually exposed to its toxic potential. The biological tension between oxygen as indispensable and oxygen as poisonous points toward unusual solutions found in some animals. Symbiotic relationships that shift the burden of metabolism onto microbial partners. In these associations, the host may circumvent part of the oxidative challenge by relying on organisms better suited to oxygen-free conditions, raising questions about where the boundary between self and symbiont should be drawn. The absence of oxygen does not necessarily mean the absence of complexity. In many environments where molecular oxygen is scarce or entirely excluded, animals persist not through their own biochemistry alone, but through partnerships with microorganisms that supply the missing metabolic functions. These symbiotic arrangements represent one of evolution's most effective solutions to the oxygen paradox. By outsourcing parts of energy metabolism to microbes, animals extend their ecological range into zones where independent survival would be impossible. One of the clearest examples comes from gutless worms of the genus Olavius. These small annelids inhabit marine sediments rich in hydrogen sulfide and devoid of oxygen. Unlike most worms, they lack a digestive system altogether. Instead, they harbor dense populations of bacterial symbionts beneath their cuticle. These microbes include both sulfate-reducing bacteria and sulfide-oxidizing bacteria, which together create a self-contained energy cycle. The host worm derives nutrition from bacterial metabolism, while the bacteria gain a stable habitat. The worm's own cellular machinery does not rely on oxygen. Instead, it benefits indirectly from microbial processing of reduced compounds in its environment. Similar strategies are observed in deep-sea clams of the family Lucinidae. These bivalves, first described in the 19th century, but only later understood in biochemical detail, house sulfur-oxidizing bacteria within specialized gill cells called bacteriocytes. The bacteria oxidize sulfide to sulfate, fixing carbon in the process and providing organic compounds to the host. The clam, in return, supplies the bacteria with access to sulfide from sediment and with protective housing. The success of this arrangement allows lucinid clams to dominate habitats where oxygen levels are negligible and sulfide concentrations are high, niches that would otherwise exclude most animals. In some cases, the partnership involves organelles within the host cells themselves. Hydrogenosomes, discovered in the 1970s in anaerobic protists such as Trichomonas vaginalis, have also been identified in several metazoans. These organelles, derived from mitochondria, generate ATP without oxygen by producing hydrogen gas as a byproduct. Their efficiency is low compared to mitochondria, but they are sufficient to sustain small-bodied animals in anoxic conditions. The presence of hydrogenosomes in Loriciferans from Mediterranean hypersaline basins suggests that entire animal phyla can base their physiology on symbiosis between modified organelles and microbial partners. Even more reduced are mitosomes, found in parasites like Henneguya Salminicola. Mitosomes perform no role in energy metabolism but retain functions in iron, sulfur cluster assembly, a requirement for enzymatic activity across many pathways. The absence of oxidative phosphorylation in such organisms implies that energy production may be outsourced entirely, either to anaerobic biochemical routes or to direct uptake from host cells. These arrangements blur the line between autonomy and dependency, as the host animal's survival is inseparable from the metabolic activities of microbes or from remnants of mitochondria reshaped for oxygen-free life. The biochemical exchanges in these symbioses are often highly specialized. Sulfur-oxidizing bacteria provide reduced carbon compounds, methanogens consume hydrogen produced during fermentation, and acetogenic bacteria recycle waste products into usable substrates. In many cases, these microbial communities form consortia within the animal, establishing miniature ecosystems that parallel larger environmental cycles. The host's role is to provide stability and structure, while the microbes supply metabolic versatility. Such partnerships have profound evolutionary implications. They suggest that the boundary between animal and microbe is more fluid than once assumed. Animals adapted to oxygen-free environments may be best understood not as independent organisms, but as holobionts, integrated systems of host and symbionts functioning as a unit. The genetic and metabolic diversity of the microbial partners compensates for the narrow physiological limits of the host. From this perspective, the capacity of animals to colonize anoxic habitats is not evidence of intrinsic tolerance, but of ecological collaboration. Symbiosis without oxygen also highlights the contingent nature of evolutionary solutions. While humans rely absolutely on mitochondria and oxygen, other animals demonstrate that life can proceed along entirely different trajectories. The metabolic core of the cell, often portrayed as universal, is in fact negotiable, shaped by environmental pressures and ecological opportunities. These deviations underscore the adaptability of eukaryotic biology and caution against assuming that the oxygen-dependent model is the only possible template for animal life. The ecological reach of such organisms is extensive. Gutless worms shape sediment chemistry by channeling sulfide into bacterial pathways, altering nutrient cycles on the seafloor. Lucernid clams influence carbon and sulfur dynamics in seagrass beds, contributing to ecosystem productivity. Even parasitic cnidarians, despite their reduction, illustrate how symbiosis enables persistence in oxygen-free niches. These examples demonstrate that animals and microbes co-evolve metabolic interdependencies that reshape both physiology and environment. Yet questions remain unresolved. How stable are these partnerships across evolutionary time? What selective pressures maintain the fidelity of symbionts, preventing them from drifting towards selfish exploitation? To what extent are the genomes of host animals shaped by reliance on microbial metabolism? Comparative genomics and single-cell transcriptomics are beginning to address these issues, revealing gene transfers between microbes and hosts that suggest long-term integration. The full picture of how deeply symbiosis reconfigures animal physiology in oxygen-free contexts is only beginning to emerge. The presence of animals sustained by microbial partners raises further questions about the role oxygen played in enabling large, complex organisms. If symbiosis allows some to bypass oxygen entirely, why did most lineages embrace it so completely? The answer lies not only in the advantages oxygen confers, but also in the evolutionary trajectory that linked complexity to energy throughput. To grasp why oxygen became inseparable from the evolution of size, mobility, and intelligence, one must turn to the broader narrative of how oxygen reshaped the possibilities of multicellular life. The history of multicellular life cannot be disentangled from the rising levels of oxygen in Earth's oceans and atmosphere. Oxygen did more than provide a new electron acceptor. It created an energetic threshold that made large size, active movement, and complex nervous systems possible. The difference between anaerobic and aerobic metabolism is not incremental but exponential. The leap from 2 molecules of ATP per glucose through fermentation to more than 30 through oxidative phosphorylation provided the surplus necessary for organisms to construct tissues beyond the scale of microscopic forms. This surplus created evolutionary possibilities that would otherwise have been inaccessible. The fossil record of the Ediacaran and Cambrian periods illustrates this transformation vividly. Around 600 million years ago, oxygen concentrations in seawater appear to have risen above 10% of present atmospheric levels, as indicated by chromium isotope ratios and the disappearance of certain redox-sensitive minerals from marine sediments. This geochemical shift coincides with the first appearance of large, soft-bodied organisms preserved in Ediacaran rocks in Newfoundland, Namibia, and Australia. These organisms, some exceeding a meter in length, represent a clear departure from microbial mats and microscopic protozoa that dominated earlier epochs. Their size alone required diffusion of oxygen through tissues or, more likely, the evolution of specialized structures for gas exchange. The Cambrian explosion, beginning around 541 million years ago, further highlights the role of oxygen. Within a relatively short geological interval, arthropods, mollusks, chordates, and other major lineages diversified, many possessing hard skeletons, complex eyes, and predatory behaviors. Such innovations demanded high energy throughput. Active predation requires rapid muscle contraction, which in turn depends on abundant ATP supplies. The development of vision, particularly compound eyes and image-forming lenses, requires dense neuronal networks with continuous oxygen delivery. The expansion of ecological interactions, predation, burrowing, swimming, was contingent on oxygen providing the energy substrate for complexity. The connection between oxygen and nervous system evolution is particularly striking. Nervous systems function as information processing networks, transmitting electrical impulses at high speeds. This requires constant maintenance of ionic gradients, which consume vast amounts of energy relative to tissue mass. Comparative physiology reveals that brains are among the most metabolically expensive tissues in the animal kingdom. The expansion of cephalopod nervous systems in mollusks and the vertebrate brain in chordates correlates with oxygen-rich environments that could support such energy-intensive structures. Oxygen, in this sense, was not merely a permissive factor but a driving force in the trajectory toward intelligence and behavioral sophistication. At the molecular level, oxygen also facilitated the evolution of collagen, the most abundant structural protein in animals. Collagen synthesis requires hydroxylation of proline and lysine residues, a reaction that depends on oxygen and ascorbate as cofactors. Without collagen, the construction of connective tissues, skeletons, and complex body plans would not have been possible. The rise of oxygen therefore intersected with both metabolism and structural biochemistry, enabling not only energy-rich physiology, but also the physical frameworks of multicellular organization. Yet this evolutionary embrace of oxygen carried with it a rigid dependency. Once lineages invested in oxygen-dependent pathways, they became locked into them. The integration of mitochondria into eukaryotic cells was a pivotal moment. These organelles specialized in aerobic respiration, and over time, their genomes were reduced, with most functions transferred to the host nucleus. The irreversible incorporation of mitochondria meant that eukaryotic complexity became tied to oxygen availability. Unlike bacteria or archaea, which could switch between electron acceptors depending on conditions, multicellular animals lost such flexibility. Evolution traded versatility for power. This trade-off explains the contrast observed today between animals that require continuous oxygen supply and microorganisms or small invertebrates that can persist without it. The same energetic intensity that allowed for mobility, predation, and intelligence also created absolute dependency. Large body size compounds this constraint. Diffusion alone cannot supply oxygen efficiently to tissues beyond a few millimeters thick, necessitating circulatory and respiratory systems that maintain constant delivery. Such infrastructure commits the organism to oxygen as a non-negotiable requirement. The evolutionary narrative also includes fluctuations in oxygen that shaped biodiversity. During the Permian period, around 250 million years ago, atmospheric oxygen may have fallen to levels as low as 12%. This decline coincided with the largest mass extinction in Earth's history, suggesting that many oxygen-dependent organisms could not withstand reduced availability. Later increases during the Carboniferous, when oxygen levels exceeded 30%, supported giant insects and amphibians, their body size made possible by high atmospheric concentrations. These oscillations demonstrate how closely biological potential has been tied to oxygen throughout geological time. Even within modern ecosystems, the link persists. Coral reefs, rainforests, and other biodiversity hotspots occur in environments with abundant oxygenation, supporting high metabolic rates and complex interactions. In contrast, oxygen-depleted zones of oceans, known as oxygen minimum zones, host reduced diversity and simpler food webs. The global distribution of life today reflects the long evolutionary dependence on oxygen as both resource and constraint. The paradox remains that oxygen both enabled and limited the course of evolution. It allowed organisms to achieve levels of complexity unattainable in its absence, but it also entrenched vulnerability. Organisms that rely on oxygen cannot readily abandon it, and their survival depends on environments where it remains available. This dependency raises pressing questions when considering resilience, extinction risk, and the potential for alternative life strategies beyond Earth. The limits imposed by oxygen dependency become most evident when considering how some lineages retained capacities for survival without it, offering insights into what oxygen-based complexity cannot achieve. The persistence of animals and microbes in oxygen-free environments provides a window into resilience that transcends human physiology. By studying species that endure anoxia, biologists uncover strategies that not only expand our understanding of evolutionary adaptation, but also offer potential applications in medicine, biotechnology, and astrobiology. The survivors demonstrate that the absence of oxygen need not equate to the cessation of life, but rather to a shift in the balance of metabolism, physiology, and ecological interaction. One of the most direct lessons concerns metabolic depression. The painted turtle, carp, rotifers, and tardigrades show that the ability to lower energy expenditure radically increases tolerance to oxygen deprivation. In medical research, this insight has inspired efforts to induce hypometabolic states in humans. Experiments at the University of Minnesota and other institutions have examined suspended animation techniques, cooling the body and reducing cellular activity to extend the window for surgical interventions or trauma recovery. The principle echoes what overwintering turtles achieve naturally: survival not through continuous energy production, but through slowing life processes to a level compatible with restricted oxygen availability. Organ transplantation provides another context where these lessons resonate. Once removed from the donor, organs are deprived of circulating oxygen and begin to accumulate damage from ischemia and reperfusion. Researchers have investigated whether protective strategies drawn from anoxia-tolerant species can extend organ viability. For example, turtle neurons exposed to anoxia avoid excitotoxicity by releasing inhibitory neurotransmitters, a mechanism that inspired studies on pharmacological agents to protect human neurons during strokes or cardiac arrest. Similarly, the remarkable stability of tardigrade proteins in desiccated states has encouraged the exploration of novel cryoprotectants and stabilizing agents for preserving tissues and organs at low temperatures. The ability of some animals to buffer or eliminate metabolic byproducts also offers biomedical parallels. Carp convert anaerobic end products into ethanol, which diffuses harmlessly away, while turtles sequester lactate in their shells. These solutions are unavailable to mammals, but they demonstrate alternative strategies for managing acidosis. In clinical scenarios such as cardiac arrest, where lactic acidosis contributes to tissue injury, mimicking or modifying these pathways could prove beneficial. Scientists have begun probing whether metabolic engineering or synthetic biology might one day provide mammalian cells with improved tolerance to acid buildup, extending survival during oxygen deprivation. Another lesson emerges from symbiosis. Worms, clams, and parasites that rely on microbial partners highlight the possibility of outsourcing metabolic functions. While humans cannot easily establish such partnerships, understanding how hosts integrate microbial metabolism sheds light on the flexibility of biological systems. In oncology, for instance, tumors often create hypoxic microenvironments, forcing cells to shift to anaerobic metabolism. Insights from symbiotic systems where oxygen is absent may clarify how cells can persist under hypoxia and suggest new therapeutic targets to disrupt malignant growth. The study of anoxia-tolerant organisms also informs our understanding of aging. Oxidative stress, generated by reactive oxygen species, contributes to cellular senescence and tissue decline. Animals that avoid oxygen altogether, or that enter dormant states where oxygen demand ceases, escape many of the damaging effects of ROS. Deltoid rotifers, capable of surviving decades of desiccation and anoxia, show limited accumulation of oxidative lesions compared to continuously aerobic species. The implication is that avoiding or minimizing oxygen metabolism may confer longevity benefits, though at the cost of reduced activity and complexity. For humans, whose survival depends on oxygen, this balance cannot be shifted wholesale, but the underlying principles may guide interventions aimed at reducing oxidative damage. In the context of human exploration and survival in extreme environments, these lessons take on added urgency. Deep sea, polar, and space missions all confront the problem of oxygen scarcity or instability. Learning from organisms that can endure without oxygen suggests avenues for developing countermeasures, whether through metabolic suppression, protective proteins, or novel biotechnologies, that might allow humans to extend endurance in hazardous conditions. Even if independence from oxygen is unattainable, greater tolerance could provide critical advantages. Yet there are limits to how far such lessons can be applied. The complexity of the human brain, heart, and muscles sets thresholds that cannot easily be relaxed without impairing function. Metabolic suppression in humans risks irreversible loss of consciousness or vital organ damage, outcomes incompatible with survival in everyday ecological or social contexts. The strategies of carp or tardigrades work in part because these species can endure long periods of inactivity or suspended states, conditions that would compromise human survival outside of controlled environments. The survivors teach us what is possible biologically, but also highlight what is constrained by evolutionary commitments. The broader perspective is that oxygen-free animals and microbes demonstrate the plasticity of life. Evolution does not follow a single path, but explores multiple solutions depending on environmental pressures. Where oxygen was abundant, complexity blossomed. Where it was absent, resilience and efficiency took precedence. Both strategies are valid, and both reveal truths about the nature of living systems. The survivors remind us that oxygen is not universal law, but one trajectory among many, and that adaptability often lies in pathways least expected. The implications extend beyond Earth. If organisms can thrive without oxygen here, then oxygen may not be a prerequisite for life elsewhere. The very existence of anoxia-tolerant and anoxia-independent species forces us to reconsider assumptions about habitability on other planets and moons. To understand what such lessons mean for the search for extraterrestrial life requires examining how Earth's oxygen story intersects with astrobiology, and whether oxygen truly serves as a reliable beacon of life across the cosmos. The recognition that life on Earth can persist entirely without oxygen reshapes the way scientists search for biology beyond our planet. For decades, oxygen in an exoplanet's atmosphere has been treated as a potential biosignature, its presence interpreted as evidence of photosynthesis and, by extension, of living processes. Yet the discovery of animals, protists, and microbial consortia that thrive permanently in anoxic conditions complicates this assumption. If oxygen is neither necessary nor universal, then the parameters for habitability must extend well beyond the boundaries set by terrestrial aerobic life. Astrobiology has been particularly shaped by comparisons to Earth's extremophiles. Subglacial lakes in Antarctica, brine pools in the Mediterranean, and hydrothermal vents along mid-ocean ridges provide analogues for environments that may exist on icy moons or subsurface oceans elsewhere in the solar system. Jupiter's moon Europa, with its saltwater ocean beneath kilometers of ice, and Saturn's moon Enceladus, with its plumes of hydrogen and organic molecules detected by the Cassini spacecraft, represent prime candidates. In such settings, oxygen may be scarce or absent, yet life based on anaerobic metabolisms, sulfate reduction, methanogenesis, or hydrogen oxidation, could still flourish. The metabolic strategies observed in Earth's oxygen-free organisms illustrate what might be possible in these alien habitats. The chemical signatures of life in anoxic systems differ sharply from those of oxygen-rich ecosystems. Instead of oxygen accumulation, one might expect methane, hydrogen sulfide, or organic acids to mark biological activity. The challenge is distinguishing between abiotic and biotic sources of such compounds. Methane, for instance, can be produced by both geological processes and by methanogenic archaea. Identifying patterns such as isotopic fractionation, where biological processes preferentially use lighter isotopes, may provide a more reliable indicator. Earth's record of methane-rich atmospheres during the Archean Eon demonstrates that abundant biological activity can exist without producing oxygen as a detectable signal. Exoplanetary studies add another dimension. Telescopes such as the James Webb Space Telescope and future missions like LUVOIR and HAB-X aim to detect atmospheric compositions of distant planets. Oxygen, while striking if observed, is only one molecule among many. The recognition that life may not produce oxygen at all broadens the scope of biosignature searches to include combinations of gases in disequilibrium, such as methane and carbon dioxide coexisting in concentrations that would ordinarily react away. Such disequilibria might signal active metabolisms that do not rely on oxygen. Theoretical models also suggest that life elsewhere could adopt pathways never realized on Earth. Alternative electron acceptors such as perchlorates, detected on Mars, or sulfur compounds in volcanic environments, could sustain microbial ecosystems without oxygen. The chemistry of Titan, Saturn's largest moon, dominated by methane and hydrocarbons, raises the possibility of life forms based on entirely different solvent systems, such as liquid methane instead of water. Here, Earth's oxygen-free organisms provide a conceptual framework. They show that the absence of oxygen need not preclude complexity, only that complexity would take different biochemical forms. The debate within astrobiology reflects a broader philosophical shift. For much of the 20th century, oxygen was equated with progress and higher life. The expansion of multicellular organisms on Earth during the Proterozoic and Cambrian was closely tied to oxygen availability, reinforcing this view. Yet the persistence of life in oxygen-deprived niches demonstrates that such a perspective is parochial. Evolution did not converge on oxygen as a universal solution, but explored a spectrum of possibilities, some of which remain hidden in the anoxic depths of our own planet. This realization influences the design of exploration missions. Instruments planned for Europa Clipper or Dragonfly, the rotorcraft destined for Titan, are calibrated not to detect oxygen specifically, but to identify complex organic chemistry, redox gradients, and potential biosignature molecules. The guiding question has shifted from, is oxygen present? To, are there energy sources that biology could exploit? The experience from Earth's anoxic ecosystems indicates that life can harness gradients of hydrogen, sulfur, or carbon compounds as readily as oxygen. The implications also extend to questions of resilience. If life can adapt to oxygen-free niches on Earth, then it may also persist on planets where oxygen once existed but has since disappeared. Planetary atmospheres evolve over time, shaped by volcanic activity, solar radiation, and impacts. A world that once harbored photosynthetic organisms may later become anoxic, yet life could endure in subterranean or suboceanic refuges. The survival of microbes in Earth's deep biosphere, sealed away from oxygen for millions of years, illustrates the plausibility of such long-term persistence. These considerations complicate how we interpret oxygen when it is detected. While its presence may suggest photosynthetic activity, oxygen can also accumulate through abiotic processes, such as the photodissociation of water vapor under intense ultraviolet radiation. Conversely, its absence does not negate the possibility of life. The lesson from Earth's anoxic survivors is that biology is more versatile than a single atmospheric signal. To equate life with oxygen is to risk overlooking forms that operate under entirely different chemical regimes. The broader search for alien life thus hinges on a delicate balance, recognizing the extraordinary potential of oxygen-based metabolisms while avoiding the assumption that they are the only viable pathway. As telescopes probe distant worlds and spacecraft explore icy moons, the challenge will be to refine our criteria for detection in light of Earth's diverse examples. The capacity of life to thrive without oxygen expands the boundaries of what might be found elsewhere, demanding that the search remain open to unfamiliar chemistries and unexpected adaptations. Yet the reflections drawn from anoxic organisms also cast light back on ourselves. If other worlds may support life without oxygen, the dependence of humans and other complex animals on it appears less as a universal rule and more as a contingent outcome of evolutionary history. This perspective invites a closer examination of why our species, unlike many of Earth's survivors, has no alternative path, and what this dependency means for our biological limits. Human physiology is constructed around oxygen with a rigidity unmatched by most other organisms. Every system, circulatory, respiratory, neurological, muscular, operates on the assumption of continuous delivery. While certain animals have demonstrated ways to tolerate or abandon oxygen, the human body remains fixed in its reliance. The margins for survival are narrow, measured in minutes of deprivation before irreversible damage accumulates. This constraint defines not only our biology but also the boundaries of our endurance, adaptation, and technological interventions. The mechanisms of this dependency are most evident in cellular metabolism. In humans, oxidative phosphorylation accounts for more than 90% of ATP production under normal conditions. Skeletal muscles, the heart, and the brain all consume oxygen at rates proportional to their activity, with little capacity to shift toward alternative electron acceptors. The limited ATP yield from glycolysis is insufficient to sustain these tissues beyond transient episodes of hypoxia. During extreme exertion, when oxygen delivery cannot meet demand, muscles rely on anaerobic glycolysis briefly, producing lactate, yet this pathway cannot compensate for systemic anoxia. The body is designed not for flexibility, but for throughput. Training can stretch these margins, though never eliminate them. Free divers illustrate the extent of human tolerance. Through years of conditioning, they suppress reflexes, optimize oxygen storage in hemoglobin and myoglobin, and slow heart rate to conserve supply. Records exceed 11 minutes of voluntary apnea, But even in these cases, oxygen is never absent. Stores in blood and tissues are simply rationed more efficiently. The limit remains strict. Once hemoglobin desaturates and tissue oxygen levels fall, syncope and injury follow rapidly. Adaptation can extend tolerance, but cannot change the fundamental requirement. High-altitude populations provide another case of extended but constrained adaptation. In the Andes, Tibet, and Ethiopian highlands, humans live at altitudes where oxygen availability is reduced by up to 40% compared with sea level. Genetic studies reveal distinct solutions. Tibetans display increased ventilation and nitric oxide production, enhancing oxygen delivery without elevating hemoglobin excessively. Andeans compensate by raising hemoglobin concentrations, improving oxygen transport at the risk of increased blood viscosity. Ethiopians exhibit a yet different strategy, maintaining near sea level hemoglobin with mechanisms still under investigation. Each represents an adjustment to scarcity, but none constitutes independence from oxygen. The reliance persists. The adaptations only shift the thresholds of tolerance. Medical interventions attempt to address this rigidity through technology. Mechanical ventilation, extracorporeal membrane oxygenation, and hyperbaric chambers provide artificial means of sustaining or enhancing oxygen delivery. These methods under the absence of intrinsic alternatives. Rather than switching to new metabolic pathways, the human body requires external supplementation when natural delivery falters. Organ preservation research continues to explore whether temporary hypometabolic states can be induced, drawing inspiration from anoxia-tolerant species, but even under laboratory conditions, complete independence remains unattainable. The paradox of oxygen becomes evident in the context of aging and disease. While indispensable for energy production, oxygen also generates reactive oxygen species that contribute to cellular damage. Antioxidant defenses mitigate, but do not abolish this effect. Over decades, oxidative stress accumulates in mitochondrial DNA, proteins, and lipids, contributing to senescence. Thus, the very element that sustains life also imposes a cost in the form of gradual decline. Humans exist within this paradox, dependent on oxygen for every function, yet subject to its corrosive byproducts across a lifespan. From an evolutionary standpoint, the inflexibility of human oxygen dependence reflects the trajectory of complexity. The expansion of the neocortex, the rise of endurance locomotion, and the energetic demands of social behaviors required consistent and abundant energy. Aerobic metabolism provided that energy, but at the price of eliminating fallback options. Evolution locked the human lineage into a single strategy: high performance tethered to oxygen, with little room for alternatives. This limit has consequences for how humans confront environments beyond Earth. Space missions, deep-sea exploration, and polar expeditions all depend on continuous artificial oxygen supplies. Unlike extremophiles that retreat into dormancy or shift to different electron acceptors, humans must transport their atmosphere with them. Oxygen tanks, regenerative life support systems, and controlled habitats are extensions of our physiology, necessary prosthetics for survival in environments that do not match our evolutionary constraints. The requirement defines both the fragility and the ingenuity of human presence in hostile settings. Yet the existence of organisms that live without oxygen compels reflection on the narrowness of this human limit. Their presence demonstrates that life does not conform to a universal dependency, and that alternative strategies remain viable under the right ecological conditions. For humans, whose biology has been shaped by oxygen-rich histories, such alternatives lie beyond reach. What remains open is the question of whether future science, through biotechnology, synthetic biology, or genetic modification, might one day extend the boundaries of tolerance, offering glimpses of what independence from oxygen could mean. The human condition, then, is one of captivity within an atmosphere that both sustains and erodes. Oxygen empowers cognition, motion, and culture, but its absence reveals the immediacy of mortality. Other organisms show that the rules of life are broader, that resilience and survival take forms unfamiliar to our physiology. The study of those forms forces recognition of both the contingency of our dependence and the potential diversity of life beyond it, a recognition that unsettles the notion that our way of living defines the boundaries of what biology can achieve.